Hello everyone, welcome back to Fundamental of Biochemistry series. This is episode 6 and in this episode we are going to discuss about heteropolysaccharide. In last episode we discussed the polysaccharide and in detail about the homopolysaccharide. So in this episode let's discuss about heteropolysaccharide are composed of two or more different type of monosaccharides or derivatives which are in repeating or non-repeating patterns. They are classified if they are it also found in classify on the basis of their found in animal and connective tissue. They are called as a mucopolysaccharide. If it found in plant cell wall and its secretion, then we called as a plant heteropolysaccharide. It present in algae, then we called as a algal heteropolysaccharide. It are present in bacterial cell wall structure, capsule, outer membrane, then we called as a microbial heteropolysaccharide, and it also present in a animal, then animal derived heteropolysaccharide, right? So let's discuss each one in detail. So first one is a glycosaminoglycans. It means GAGs. It is also called as a mucopolysaccharide, which is repeating sugar uh, of the disaccharide unit of amino sugar as well as the uronic acid. It is also associated with the sulfated group. It is found mainly in animal connective tissue and body fluids. These are the various example of the mucopolysaccharide: heparin, hyaluronic acid, chondroitin sulfate, keratin sulfate, heparin sulfate. Then second is in present in plant. They are composed of different sugar units found in plant cell wall and its secretion. Examples are pectin, gums, and hemicellulose. In algal, they are extracted from the algal. Mainly, they are a brown algae that is use agar agar powder or thickening agent that will use in a various industry. So here example of algal heteropolysaccharide are agar agar, carrageenan, alginic acid, alginate, and so on. Bacterial heteropolysaccharide are found in cell wall, capsule, and multiple sugar types also present. So peptidoglycan that is present on cell wall, bacterial capsule that is also some capsulated organism which contain the capsule that is responsible for the virulence factor, as well as lipopolysaccharide which is found on the gram negative bacteria, and that is O antigen chain is present. Then animal derived heteropolysaccharide example is blood group substance which is ABO antigen that is made up of galactose, N-acetyl glucosamine, N-acetyl glucosamine, and fructose. These are the chain that are present in RBC on the surface of RBC cell, and that is a animal derived heteropolysaccharide, right? So these are the properties of heteropolysaccharide, which are multiple sugars are present, and that's why it is known as heteropolysaccharide. It is a very complex structure. It may be branch or may be linear, and it is repeating unit of the disaccharide unit. Usually they are acidic in nature because it contain the negative charge or sulfate group is present. That's why it is usually acidic in nature. It has a very water binding capacity. They are hydrophilic and can certain large amount of retain the water. They are viscous in solution, high molecular weight. Viscous gel-like solution is present. They also play important role in structural component in connective tissue, cartilage, skin, and extracellular matrix. It does not give this much function. It is also involved in provide a lubricant or protection of the tissue against the various cushioning, and etc. It is also have a biological specificity, composition, and structure determine the biological function. uh, so these are the properties of the heteropolysaccharide. Let's discuss some examples of the heteropolysaccharide, in which we first discuss about the hyaluronic acid. It is found in higher animal and component of various tissue. You can see here this is skin, vitreous body of the eye, umbilical cord, synovial fluid of the joint, and some bacteria. Has a high viscous substance. That's why it's categorized in the mucopolysaccharide. This is the molecular weight of the hyaluronic acid. It's acidic in nature, and which contain the glucuronic acid, N-acetyl glucosamine, as uronic acid. It acts as a lubricant and biological cement in the joint. Right? Hyaluronic acid is split by hyaluronidase enzymes. So some enzymes which are present or secreted by the some bacteria and which release the hyaluronidase enzyme and which break down the hyaluronic acid and which are success for spreading the infection. That's why are also called as a spreading factor. It is a straight chain polymer of this structure of D-glucuronic acid and N-acetyl D-glucosamine. So this is the structure of hyaluronic acid. Let's give the some point regarding the hyaluronidase enzyme. So we also already give that that's hyaluronic acid which is responsible for give the cement factor between the cell which is present on extracellular matrix, right? So it will connect the cell each other by hyaluronic acid. But some organism which are produce the hyaluronidase enzyme like a streptococcus, staphylococcus aureus, clostridium, and streptococcus pneumoniae. These organisms which release and that break down this cement material which are present between the cell. Right, so that will this is the mechanism, and that hyaluronidase enzyme which will break down cleave beta 1 for glycosidic bond between this sugar molecule and which converted into small structure. And here you can see this is a complex structure by due to the help of hyaluronic acid. After the attack on hyaluronidase enzyme, you can see that dissolve the intracellular cement material which hold the cell, right? So it increase the tissue permeability. When we can said that pathological significance which are this enzyme called as a spreading factor because it allow the bacteria and the toxin and into the deeper tissue and which spread rapidly through the connective tissue and along tissue the planes. It contributes the cellulitis, necrotic fasciitis, gas gangrene, and abscesses, so on. Let's discuss a second example, that is chondroitin sulfate. Chondroitin sulfate are also mucopolysaccharide. It also called as a glycosaminoglycans. It found is you can see here cartilage, bone, skin, heart valve, and tendons. It contributes the tissue ability to withstand force and provide the flexibility. Due to our bones are flexible due to this chondroitin sulfate. There are three types of chondroitin sulfate: A, B, and C. These all these heteropolysaccharide which contain the sulfate group you can see here. Third example, that is heparin. First we discuss about the chondroitin sulfate, heparin, and hyaluronic acid. And next let's discuss about heparin. That heparin is also heteropolysaccharide highly sulfated group present in the structure. Disaccharide repeating disaccharide unit is present, that is composition is uronic acid and amino acid sugar, which is acidic in nature because repeating amino sugar uronic acid and this mostly having the sulfated group. That's why it, you can see here this is a negative charge, that's why it acidic in nature. Here you can see that is joined by alternate alpha 1 for glycosidic linkage heavily sulfate group in their structure. This is the molecular weight 17,000 to 20,000 molecular weight of the heparin. What is the main function of the heparin, how it will produce? So heparin is present into the mast cell which will secrete outside along with the histamine. It is abundant in perivascular connective tissue where mast cells are dense. For example, arterial wall, blood, lung, liver, kidney, and spleen. Other it main function is the heparin, that is act as an anticoagulant agent. How? Let's discuss. You can see there this is the heparin structure. In you can already know that there is a hemostasis process is there in which normally blood are going to clot during the blood flowing. At that time that there is a two pathway are they run, common pathway run, and along with that during that pathway there is activation of antithrombin and that will convert it into factor 10 and thrombin that ultimately converted into insoluble fibrin, but that make the coagulation this pathway. but heparin is a different function that will not allow to clot the blood, so that's why it's called as a anticoagulant agent. How that will act? That will bind with the antithrombin 3 and as well as thrombin factor. That's why it will not activate further, and it heparin act as a anticoagulant agent. So you can this is uh, about the heparin. Let's discuss about the another example, that is the agar-agar. Agar-agar are a heteromucopolysaccharide acidic once again in nature that are extracted from the cell wall of the red algae. They are composition. Agar-agar is made up of two components. That is, one is agarose and a second is agaropectin. Agarose is the main major component, and that are responsible for gel or solidifying fine. This is the repeating D-galactose 3,6 N-hydro L-galactose, which is the link by beta 1,4 and alpha 1,3 glycosidic bond. This is the structure of the agarose. Sorry, agar-agar. Then when we discuss about the physical property, it is called as a thermal hysteresis, because agar-agar, this is a unique feature which are melt at above around 85 degrees Celsius, and it become a clear solution or clear gel. But when we cool down from melting a point to it, melting agar to cool at 45 to 50 degree, it converted into molten form, in which generally may pour plate during the pour plate technique, this molten agar is used. So at that time temperature is 45 to 50 degrees Celsius, and that will this is the normal or temperature for inoculation of bacteria, blood, a serum, antibiotic, and so on. This is the ideal temperature for pouring plate technique. Then after cooling, the 32 to 40 degrees Celsius is settling point known as, it is solidified the totally at the room temperature. So this agar-agar is used as a solidifying agent. So now let's discuss how this agar-agar is obtained. So we already discussed that is obtained from a cell wall of the red alga. Which red alga? So is Giladium and Gracilaria are majorly used, and that are produce a certain only in red algae. This is the example of red algae, that is Rhodophyta, and this is Gileadia and Gracilia species which are produce this algae, which are insoluble in cold water but soluble in the hot water. And they are thermoversatile versible gel. So as we discussed, melting point it is 85 degree, molten form at 45 to 50 degree, and gelling point that is 30 to 40 degree. So from clear solution to molten form, molten to completely solidifying pattern, you can observe in a same kind of agar but different temperature. Where it is used as a solidifying agent in desert, food industry, vegetarian gelatin substance. It also used in a textile industry by seizing, printing, etc. It majorly it used in a microbiological laboratory in a solidifying agent in microbiological media. It used in a dairy product for stabilizer and as a thickener. So this is the whole thing about the agar-agar. Let's discuss about the another heteropolysaccharide, that is peptidoglycan. Peptidoglycan are present on the cell wall structure of the bacteria. So it made up of, and you can see here, this is the figure, N-acetyl glucosamine and it repeating unit of N-acetyl muramic acid. You can see here. So N-acetyl glucosamine which link with the N-acetyl muramic acid, and N-acetyl muramic acid which cross-link with the short peptide amino sugars are present. So it provide, what is the important of this peptidoglycan heteropolysaccharide to the bacteria? It provide the strength, rigidity to the bacterial cell wall. That's why this bacteria maintain their shape. It prevent from the osmotic lysis. So this is the peptidoglycan. Let's discuss about another example, that is pectin. Pectin also heteropolysaccharide that is present in a plant. That's why plant polysaccharide, major component of the pectin, that is D-galacturonic acid residue linked by alpha 1 for glycosidic bond. It also contains various this rhamnose, arabinose, galactose in varying amount of this sugars is present. Location mainly where this pectin is present, mainly in middle lamina. You can see here, this is a middle lamina between two cells, that is a primary cell wall of the plant. Function, it provides the adhesion and structural strength between the plant cell. Very important, it is a natural plant polymer and essential for plant structure and food industry. Especially presence of sugar in the acid, widely used as a gelating agent, gelling agent in jams and jellies. So you can see that pectin are nature's glue that holds plant cell together, and as well as it also useful in food industry and delight to our food. So these are the various also heteropolysaccharide resin gum that is also produced by the plant, secreted by the plant. These are the also heteropolysaccharide. These are the various MCQ regarding the heteropolysaccharide that you have to solve it and boost your exam, prepare your exam. Thank you very much for attention this episode.